So, it's 5:05. I think that we can start. We already have a very high presence of many people, 29 in total. So I think that's very good. And they're still getting connected to the webinar. So I'll start with hello, everyone, and good afternoon. Welcome to our webinar. We are thrilled to have you join us today. My name is Emmanuel, and I'll be your host in today's session. We have the honor of having Dr. Mark Hall as the speaker, who will share his insights and expertise with you. But before we get started, I'd like to go over a few housekeeping items. Please make sure that your microphones are muted to minimize any background noise. We encourage you to ask any questions throughout the presentation using the chat function, and we will address them during the Q&A session at the end, okay? So I'm gonna talk a little bit about Dr. Mark Hall. He's a professor and associate head of the Department of Biochemistry at Purdue University. In 1994, he completed his bachelor's in biology at Trinity College. And in 1998, he earned a PhD in biology at the University of North Carolina under the mentorship of Dr. Stephen Matson. Dr. Hall has received the Intramural Research Training Award in 1998, which allowed him to pursue a postdoctoral fellowship under the mentorship of Dr. Thomas Kunkel at the Laboratory of Molecular Genetics at the National Institute of Environmental Health and Science. Later, in 2001, he worked as a postdoctoral research associate in the Department of Biochemistry and Biophysics at the University of North Carolina. Dr. Mark Hall, obviously, as many other of his achievements, has received several awards and honors, the most recent being in the 2023, the induction into the Book of Great Teachers at Purdue University. His research interests include the identification of new candidate antifungal drug targets, focusing on the pathogenic yeast candida, and also the characterization of phosphatases and kinases that play important roles in regulating fungal-specific aspects of the cell division cycle, and also the development of tools that combine inducible protein degradation technologies with in vivo infection models or proteomics to identify new drug targets and characterize proteins in fungal pathogens. His work has led to several publications over the past 8 years. So, without further ado, and we have the honor to have Dr. Mark Hall here with us to present his in this webinar. So, without further ado, please, Dr. Hall, I give you the floor.
Thank you very much, Emmanuel, for the introduction. That was very kind, and thank you to the Puerto Rico Society of Microbiology for the invitation to talk to you all. It's a real pleasure to be here. See if I can get my screen shared. So hopefully that looks okay. So yeah, I'm excited to tell you a little bit about our work on trying to contribute to the challenge of new antifungal drug discovery. This is actually a new area for my lab within the past few years, and so we're still learning a lot. I think for a lot of people in the general public, their awareness of fungal pathogens may be limited to things like the HBO series The Last of Us. It's not something that you hear a lot about in the popular media. This show actually was good in raising awareness for fungal pathogens, and although it is a highly fictionalized story of fungal pathogens, this is a real problem that we have as a society, and it's a growing problem. So a recent estimate came out this year that invasive fungal diseases kill over 3 million people around the world each year. And so to put that in perspective, that's more than the deaths from malaria and tuberculosis combined. Yet we don't hear nearly as much about fungal diseases as we do about those diseases and other infectious diseases. Fungal diseases are particularly problematic. The mortality rate for these on average is close to 50%. And part of the problem is we just don't have a lot of good drugs and therapeutic options to combat these diseases. Um, and another reason we don't hear about them so often is they're mostly diseases that affect people that are already sick with other things. So immune compromised people are the most common victims of fungal disease. And so that people taking immune suppressant drugs, it could be cancer chemotherapies, people with HIV or other major infections like COVID or influenza, or chronic conditions like diabetes, all become susceptible to invasive fungal infections because of suppressed immune systems. And I've listed here on the bottom several of the most common fungal pathogens that affect humans. And the severity of the problem was highlighted in 2022 when the World Health Organization put out their first ever report specifically on fungal pathogens. And they grouped fungal pathogens into priority groups with a critical group here that includes the three species that I just showed on the previous slide, as well as a number of other pathogens of increasing concern. And really, the point of this report for the WHO was to draw awareness to the need for increased uh, research funding to understand pathogen biology and to discover new and optimize existing therapeutics to combat these deadly diseases. So I'm going to be talking primarily about Candida albicans. And so just to give you a quick introduction about this pathogen, this is a commensal yeast. It lives on most of our bodies as part of our natural microbiome. It lives on our skin, mucous membranes, and our intestinal tract. And for people with healthy immune systems, it's not much of a problem. But when the immune system is suppressed, it can cause a variety of diseases, including superficial conditions, like as you see in these pictures here. But it also can get into the bloodstream and be disseminated throughout the body to organs. And once it gets inside the body like that, it's very challenging to deal with. And thus we have these very high mortality rates. This is the most common source of fungal infections in humans. It's also, of all pathogens, all infectious diseases, it is the fourth highest cause of hospital-acquired bloodstream infections. So it is quite a common problem that we have to deal with. One other thing I want to point out about Candida albicans is it is a dimorphic fungus. This will become important with some of the data I show later. It exists in two forms. It exists in a budding yeast form, which you can see a budding cell right here in the top left of this lower picture. But it also can transition into a hyphal form where it grows as this elongated multi-cellular structure. And here's actually a colony growing on a plate showing these radial hyphal filaments that emanate from the central colony. Both of these forms, the yeast form and the hyphal form, are essential for this organism to be pathogenic and cause disease. And so some therapeutic strategies are to target the transition between the yeast and hyphal forms, and that'll become important in my talk later. So there's a dire need for new antifungal drugs. There's really only 4 classes of FDA-approved treatments to combat invasive fungal diseases. And examples of those are shown over here on the right. The one that I want to mention is the echinocandins, because that'll play a role in my talk. These are drugs that are derived from natural products that are actually made by some fungal species, and they act by inhibiting synthesis of the fungal cell wall. And they're one of the best therapeutics that we have. But some of these therapeutics don't work against some pathogens. Pathogens are developing resistance to some of these. We actually have examples now of clinical isolates of some pathogens that are resistant to all of the existing FDA-approved antifungal drugs. So one of the major important solutions to this problem is we need to develop new antifungals that have unique targets and unique modes of action that are different than our existing therapeutics. And this is the problem that we've chosen to try to address. And so in general, there's a variety of ways to approach antifungal drug discovery. One could screen for new targets. So that is proteins or other molecules that are actually important for virulence. And once you find those proteins, you can then focus efforts on creating and developing chemicals to inhibit or modulate the function of those targets. Another approach would be to just directly screen for agents, chemicals that can impact the disease. So find things that kill fungal cells, for example. And that's actually how the echinocandins were discovered. So those were discovered as natural products produced by some Aspergillus species that had a killing effect on other fungal cells. And then another way to approach antifungal drug discovery, or any kind of drug discovery really, is once you have a target that's known, something that is important for the disease or for the virulence of the pathogen, to use structure guided design to create chemical molecules that can inhibit that target. So as an example here, the Paxlovid drug that's used or marketed by Pfizer for treating COVID was developed in this way. So I'm going to tell you two short stories today from my lab that are going to address two of these different approaches to antifungal drug discovery. The first one is going to be the development of a functional genomics platform in our lab to use in fungal pathogens for the purpose of identifying new targets for antifungal drug development. So that'll be the first part. And the second part, we'll talk about um, a particular target, the CD14 phosphatase, that my lab's been working with for years, that we believe is an attractive target for developing new antifungal drugs. And we'll talk about why that is and about our initial efforts to find inhibitors for this enzyme. Okay, so for part one, this platform we're trying to develop is based on an inducible protein degradation system called the auxin-inducible degradation system, or AID for short. This is a system that was developed by other scientists about 15 years ago. It is a tool for triggering the rapid degradation or destruction of a particular protein of interest inside cells. And it's based on the natural function of the plant hormone auxin. So in plants, auxin is a hormone that promotes plant growth and development. This diagram here shows the way that it normally acts. And so in the absence of auxin in plant cells, there's a class of transcriptional repressor proteins called the AuxIAA proteins that bind and inhibit a class of transcription factors responsible for expression of plant growth and development genes. All right, and so those genes are off. Auxin, when auxin is present or produced by the plant, and auxin is a simple small molecule. It's indole acetic acid. It looks sort of like a tryptophan amino acid. But when it's present, what it does is it creates a strong binding interface. It acts as what we call a molecular glue to bring the AuxIAA transcriptional repressors in contact with another protein, an F-box protein called tier 1. And that's a component of a ubiquitin ligase enzyme complex. And when the AuxIAA proteins are bound, they get polyubiquitinated by this enzyme. And that polyubiquitin chain is recognized by the proteasome, which is a large proteolytic complex responsible for most protein degradation in eukaryotic cells. So auxin triggers the destruction of these transcriptional repressors, and that causes activation of the ARF-dependent growth and development genes, and that's how auxin works in plants. So what these clever scientists did about, this was Kanamaki and colleagues in Japan about 15 years ago, they showed that you can actually reconstitute this process in other organisms that don't normally respond to auxin. And all you need to do that is you need to express that tier 1 F-box protein, this blue protein here, in another organism and fuse a target protein of interest genetically to the auxin binding domain from this AuxIAA protein. Okay, and so if you do those two things, the SCF ubiquitin ligase complex is so highly conserved that the plant tier 1 protein can still be assembled into it, and in the presence of auxin will recruit the auxin binding domain in your target protein, causing it to be polyubiquitinated and degraded by the proteasome. And this system has now been demonstrated to work in human cells, in a variety of research model organisms, Drosophila, C. elegans, Saccharomyces cerevisiae. It had never been used in fungal pathogens before. And so that our initial challenge was to see if we could set this system up to work in fungal pathogens. We did it first in Candida albicans, and it turns out it works beautifully in Candida albicans. And so I'm showing an example of how it works here. These are Western blots, and so we're detecting our target protein of interest. This is CDC14. It's the enzyme I'm going to talk to you about later, our favorite protein at the moment. And this is a synthetic auxin, 5-ADIA is a synthetic auxin that works with this second generation AID system. And even at low nanomolar concentrations, when we add it to cells, we can trigger the degradation of our target protein. All right, P star is just a loading control for these different samples. And then if we look at the time, we've chosen one of these concentrations, 50 nanomolar, and looked at the degradation over time. And what's really amazing about the system is how fast it acts. Within 10 minutes, we can get almost complete loss of our target protein. And so it really acts like a drug. It has the speed and efficacy that you would expect for a chemical inhibitor when you add it to a biological system. And so that gave us the idea that we might be able to, so we use this system for protein functional characterization, and that's what it's commonly used for, but it gave us the idea that we might be able to use this system as a proxy for a drug and actually screen for things that would be good drug targets. So before I get to that, let me just back up a second and show you again at the phenotypic level how well this system works. And so the idea is, can we generate phenotypes associated with loss of target protein function? So what I'm doing here is comparing the AID system to what we see when we completely delete the CD14 genes or have a mutant CD14 in the Candida albicans cell. So there's a lot of stuff on here, so I'm going to try to walk you through this. This assay that you're seeing here is what we call a spotting assay. We have several different strains of Candida albicans. Some of them have wild type, normal CD14 at the top two rows. The bottom two rows are CD14 mutant strains. They have either permanent loss of the CD14 genes or express a mutant CD14 with reduced activity. And then in the middle, we have two examples of our auxin binding domain tagged CD14 strains. So these are the ones that should be responsive to auxin when we add it. And we just grow up liquid cultures and make serial dilutions of them and spot them on agar plates with different compounds in the media and look at how they grow. One of the phenotypes that we knew from our prior work was associated with loss of CD14 function is that the cells are hypersensitive to a drug called micafungin. That's one of the echinocandins. I introduced the echinocandins in a previous slide as one of our FDA-approved drugs for fungal infections. Micafungin is one of those echinocandins. And we knew that CD14 deletion cells are hypersensitive to that. So you can see here, our wild type cells can still grow. They're slow, they're sick, it's still a stress for them, but they can grow in the presence of microfungin, but the CD14 mutants cannot. What's important here is that our auxin binding domain, our AID tagged CD14 strains can also grow just like wild type in the absence of auxin. So when they have just microfungin present. But importantly, if we add auxin, the synthetic auxin, in addition to the microfungin, now our AID tag strains can't grow anymore. They now look like the mutant strain. So the really nice thing about this is we can convert a wild type, a phenotypically wild type cell to a mutant phenotype very simply just by adding the simple chemical compound to the media. We also showed this work with another phenotype for CD14, and that is the formation of hyphal filaments, which I mentioned to you before are essential for infection. And so we're looking at hyphal colonies on a plate here. Wild type Candida albicans cells will form hyphal colonies, and this is just a zoomed in view of the individual hyphae nicely, and it's independent of auxin. CD14 mutants cannot. They're completely defective under these conditions in forming hyphae. But our AID strain, in the absence of auxin, looks like a wild type cell, forms hyphae beautifully. But in the presence of auxin, it now has the mutant phenotype. It's unable to form hyphae. So this system worked beautifully. For CD14, it worked beautifully. And it gave us the idea that we might be able to use auxin as a proxy for drug action in screening for new antifungal drug targets. So there's a lot of interest recently in a class of drugs called degrader drugs. There's two classes of them, protacs and molecular glues. they basically act in the same way, and they act like auxin. Auxin is a molecular glue. They, instead of a conventional drug, which often inhibits, say, an enzyme activity, these drugs work by targeting the target protein, recruiting it to a ubiquitin ligase for ubiquitination and degradation. So you're getting rid of a protein associated with the disease instead of inhibiting its function. And there's examples of these degrader drugs in clinical trials now for things like cancers. Um, and we wondered if we could use auxin as essentially a surrogate for a drug to screen a whole bunch of different targets in an organism to find ones that might be suitable targets for further drug development. Right. So that was the question we had. Can we use this AID system to screen for genes and fungal pathogens that would be suitable drug targets? So how would we do that? This would require generating essentially a library or a collection of strains. And I've just shown a picture to kind of represent that. But each one of these wells, you would have a different Candida albicans strain or some other fungal pathogen where a single gene of interest has been tagged with the auxin binding domain. And then all of these strains would be used in some type of infection assay, plus and minus auxin. And we would be looking for strains where when auxin was added, it blocked the infection. So instead of killing the organism in the infection, with the plus auxin condition, we would now have good survival. So anything that had that profile out of all the strains we screened would be candidates for future consideration as drug targets, either for um, degrader type drugs or maybe even normal conventional drugs. So what we need to make that happen really is a simple, quick, inexpensive infection model that would allow us to screen a whole bunch of different strains in a short amount of time for not a lot of money. And the system that we're trying right now is using an invertebrate organism, the larvae of the wax moth. And you can see a picture of that here. So this is, people use it for fishing bait. And we actually buy it from bait shop, so that they're really cheap. But what's nice is you can inject fungal cultures into them. They have an innate immune system analogous to ours, and they will succumb. If you give them enough fungal cells, they will succumb to the fungal infection and die. And there's actually a very strong correlation between things that kill the larvae and things that kill mice, which are kind of the gold standard for infections. So we wanted to see if we could use this larval infection assay with the AID system. And then anything that we got as a positive out of that, we would then further test in a more relevant mammalian model system and make sure that it had an effect on virulence in mice as well. Okay, so this is a project that's been undertaken by a PhD student in my lab, Emily Danseisen. She's a second-year PhD student, and she has set up this larval infection assay in our lab and has been optimizing it for use with the AID system, and has very recently, like as of the past couple of days, generated some really exciting data for us using our CDC14 test strain. So I'm going to show you data in a little bit that shows you we know that CDC14 is actually required for infections, but you have to take my word for it now. But what she found in this assay is that so this curve right here is a wild type Candida albicans. Sorry, it's our AID tagged CDC14 strain, just without any auxin added. And you can see that that strain kills the larvae very effectively, kills almost 100% of the larvae very rapidly. And then we have two different conditions where she has also injected, after the fungal cells, injected auxin, two different concentrations of auxin that should be, if this works in the larvae, should degrade CDC14 and increase the survival rate. And you can see that this worked very beautifully. We have a highly significant increase in survival with the additional injection of auxin, again, as a surrogate drug. So we know from this result now that inside the larval cells, this auxin-inducible degradation system can work, and the auxin can be used as a surrogate drug to block infections. So we should be able to use this now to screen other strains to find things that also, if we inhibit them, will have a therapeutic effect in blocking infections. And just to show you a comparison, didn't have all of the controls that we'll ultimately need for this in this experiment, because Emily's still kind of optimizing the conditions. But we did publish last year a study showing the same assay with wild type and CS14 deletion strains. The wild type is in black here. The CS14 gene deletions, permanent deletions, are in blue. And you can see that the effect here with auxin is very similar to the effect that we got with the permanent loss of CS14 function. So we're very excited about that the system works. And what we're going to be moving on to now, first of all, Emily's also working with one of our collaborators here to set up infection assays in mice with that same strain to see if we can also block the infection with auxin in mice. We want to know if the AID system will work in other fungal pathogens. And so Emily is currently working on Candida auris, which is an emerging Candida pathogen, somewhat similar to Candida albicans, and then a completely different type of pathogen, coccidioides, which causes a disease called valley fever. It's a desert fungus, and so it's endemic to the U.S. Southwest. And we have a collaborator in northern Arizona that we're working with on that project. And then ultimately, we want to start building these libraries where we can tag a bunch of genes and start screening for things that could be good targets for degrader or conventional drugs. So that's what we're doing on the front of screening for novel antifungal drug targets. Now I want to switch gears and talk to you about our project where we're characterizing a specific target that we already kind of know from past work is important for fungal pathogens, and that's the CS14 phosphatase. Okay, so it's the example that I showed you in the previous slides. So we're kind of going backwards here chronologically, but I wanted to present that functional genomics system first. So just as a quick reminder, kinases and phosphatases are opposing enzyme activities. They regulate protein function through reversible phosphorylation in pretty much every biological process in eukaryotic organisms. So kinases act by adding phosphate from ATP onto serine, threonine, or tyrosine side chains in proteins. Phosphatases catalyze the reverse reaction. They use water to cleave the phosphoester bond and release free phosphate and restore the original amino acid. And the addition and removal of phosphate can regulate protein function. It can activate or inactivate enzymes, change cellular localization, affect stability. It has a whole bunch of effects, but it's a way to dynamically control protein function and biological processes. And kinases have been successfully targeted therapeutically in diseases like cancers and some autoimmune diseases. Phosphatases, although they should be equally targetable for effects on these same diseases, have been difficult to develop drugs for a variety of reasons. But I'm going to show you kind of our arguments for why we think CS14 may be an exception and why it might be amenable to drug development, specifically for antifungal purposes. So what makes a good antifungal drug target? So here's kind of a list of criteria that I think about when considering if something would be a good target for drug development. So first of all, the target of interest I'm talking about is a protein here. It's usually going to be a protein, but doesn't necessarily have to be. It's got to be a protein whose function is important for the pathogen to cause disease, or what we call virulence. Second, the target should be present in diverse pathogens and its role in virulence conserved in those species. So in other words, if we develop an inhibitor for a target, it would be good if it works on multiple pathogenic species and not just a single one. Third, the protein should be specific, or the target should be somewhat specific to the pathogen. So we don't want to cause severe side effects in the host organism. And that's a term we call specificity. And then finally, the protein of the target needs to be druggable. It has to be something that we have the ability to design or create a chemical compound that can bind it specifically and modulate its function. For example, an enzyme with a well-defined catalytic site. And also, we have to be able to get the drugs to that target in vivo. So the molecules that we develop ultimately have to be bioavailable. They have to get inside pathogen cells, or at least wherever the target protein is, be able to get to that target to have the effect that we need. All right, so to keep those criteria in mind as I start talking about CD14, I will address each of those with our data for this enzyme. So just a quick background on CS14. This is a enzyme that was originally characterized in Baker's yeast Saccharomyces cerevisiae, non-pathogenic organism, but a very common model system. It's what we use to make beer and wine and bread. It's the probably the most well understood eukaryotic organism and a very common model system for studying fundamental biological processes. In that organism, it plays an essential role in completing mitosis. It's actually related evolutionarily to protein tyrosine phosphatase superfamily and to a subfamily called the dual specificity phosphatases. And finally, it's important to note that CD14 is highly conserved in the fungal kingdom, although that essential function that was originally characterized is not highly conserved. It has other roles in fungal species that are more conserved. And I'll touch on a couple of those if I have time before we finish today. Importantly, CD14 has now been shown to be required for virulence of diverse fungal pathogens. So I showed you an example in Candida albicans from the larval infection assay, but we actually got interested in this whole possibility of CD14 being an antifungal target through a collaboration with one of my colleagues at Purdue, Jinrong Xu, who studies phytopathogens. So these are pathogenic fungi that affect plants. And in these two examples here, Fusarium graminearum, this is a major pathogen of wheat and other cereal crops, and Magnaporta oryzae, a major pathogen of rice. What they found is you can delete the CD14 gene and the organisms can still grow fairly well vegetatively. But what was surprising at the time and very dramatic is they were completely non-infectious. And so you can see that here. This is what they do is they just spot a little bit of culture on the head of these wheat plants, and the wild type Fusarium spreads quickly and kills the plant. Without the CD14 gene, plant is green, happy, and healthy. And this is just a complementation control where we put back in a transgene of CD14 to demonstrate that the effect is truly CD14 specific. Same thing was observed with Magnaporta using the rice plants. Um, and so and this gave us the idea that CD14 could be an antifungal pesticide target for crops because it seems to be critically important for them to infect their host plants. And what gave us even more encouragement, so actually, oh, sorry, this is just kind of a reiteration now of what we found with Candida albicans. This is the larval infection assay again. And one thing I do want to point out with this is very important also. This plot over here on the right shows biochemical enzyme activity for a mutant CD14, which we call a hypomorph, or abbreviated HM. So you'll see that in a number of my slides, CD14HM. It's a perfectly functional CD14 enzyme. It just has about 5 to 10-fold lower catalytic activity compared to the wild type enzyme. But it's very important in our characterization of it as a virulence factor in Candida albicans. And that's you can see here in this larval infection assay. Here's the deletion strain in blue. The green line is our hypomorphic mutant. So again, it has functional CD14 and it just less activity. So what that means is we don't need to completely get rid of CD14 activity to have a therapeutic benefit in blocking infections. Even partial inhibition of CD14 activity might be enough. And so based on that positive result, we went then and looked at a mouse infection assay, again, through a collaboration here at Purdue with some colleagues in the Department of Comparative Pathobiology. And the results were even more dramatic in the mouse infection assay. And so we've actually got these same strains. We injected these same strains into mice and then looked over time at the mouse survival. And so wild type Candida albicans kills all of the mice very quickly. Our deletion strain, our hypomorphic mutant strain, and our control strains all had 100% survival. And so loss or even partial loss of CD14 function rendered Candida albicans completely avirulent in this assay, which was super exciting. And so we know from these results that with that number one criteria, we've been able to check that box. That CD14 is very important for virulence in pathogens. We know it's true in diverse fungal pathogens that affect both humans and mammals, as well as those that affect plants. And so we wanted to then look at the structure and the enzymatic properties of CD14 to address the issue of whether it could be a druggable target. And one of the things, my lab spent a long time, years, characterizing the biochemistry of CD14 and its substrate specificity. So I'm not going to show you any of that data, but I'm going to summarize some important features. And one of those is that CD14 phosphatases have a unique structure compared to other phosphatases. And so at some point in their ancient evolutionary history, the phosphatase domain of CD14 enzymes was duplicated. And so this is a fundamental feature of all CD14 enzymes. They had this tandem repeat of the dual specificity phosphatase domain. And you can see that over here. This is the ribbon diagram on the right of a crystal structure of cerevisiae CD14 enzymes showing those two domains. And what's important about that is those two domains have an interface right at the active site where the substrate binds, and they create this unique channel has one wall contributed by the N-terminal domain, which is a non-catalytic domain. It's lost the catalytic site, but it still folds in the same way. And the other wall is contributed by this catalytic domain, and it creates this binding groove for peptides that has pockets for substrate features that are very important for substrate recognition and that are unique to CDC14 that we don't find in other phosphatases. And you can see that in the crystal structure here. So first of all, even though CD14 enzymes are by evolutionary history a dual specificity phosphatase, meaning they should be able to dephosphorylate phosphoserine, threonine, and tyrosine, they're very specific for phosphoserine. They won't generally touch a substrate that has a phosphothreonine or phosphotyrosine. And then they have this pocket here that accommodates a proline, specifically the trans isomer of proline, right after the phosphoserine, and then a pocket over here for a lysine side chain. And those three features, phosphoserine, proline, and lysine, are essential for good activity of CD14 enzymes. And this gave us the idea that we ought to be able to design chemical compounds that mimic this substrate and fit very specifically in this active site, but not in the active site of other phosphatases and related enzymes. Also, very importantly, through a collaboration with another colleague at Purdue, Jen Wisecaver, and her postdoc, Amanda Pendleton, we found or showed, this is a phylogenetic analysis, CD14 is essentially invariant in the Dicaria fungal subkingdom, which has almost all known pathogens. And that's this big group of organisms right here, with the red means 100% of the species have a CD14 ortholog. Blue means none of them do, right? And so the only eukaryotic organisms actually that lack CD14 are land plants. And this was a reason, again, we were interested in as a pesticide target for crops, is that plants don't have CD14 at all. Animals do, but we have reason to believe that that is not necessarily going to be a problem for targeting for drug development. I'll touch on a couple of those points in a second. This is just another way of looking at the conservation of CD14. This is the structure, again, that's been colored based on conservation across the fungal kingdom. Blue being invariant amino acid position. So here's, again, the peptide substrate binding site right here. And you can see there's this broad swath of blue, dark blue, indicating that that binding site is basically the same in all fungal species. So again, that means that if we develop an inhibitor mimicking a substrate, it's probably going to bind to many, if not all, fungal CD14 orthologs, meaning a drug could be effective against multiple fungal pathogens. And so that checks another box in the criteria for something being a good antifungal target. So just to review then, we know that CD14 is required for fungal pathogens to infect their hosts. It has a structure and substrate specificity that's broadly conserved in fungal pathogens and is also unique to CD14, meaning we might be able to develop specific chemical inhibitors of it. It's absent from plants, and so we don't have to worry about side effects in plants. And what we know about it in mammalian organisms is that, so there's a very important study a couple of years ago in mice that knocked out the CD14 genes and showed that after birth, mice can survive and live a normal lifespan without any CD14 at all. So for the purpose of temporary, treatment of a otherwise fatal fungal infection, we expect the side effects on humans should be fairly minimal with CD14 inhibitors. But as you're going to see, we also have good reason to believe that we can specifically target fungal CD14 enzymes. Okay, so now that brings up the questions, can we actually develop inhibitors for CD14? So this gives me a chance to bring up my important connection to Puerto Rico. So I have a third-year PhD student in my lab, Kevin Velasquez Morero, who is a graduate from the University of Puerto Rico at Humacao. And his thesis project is to address this challenge, developing um, chemicals that can inhibit CD14 that might be good leads for antifungal drugs. So one of the things that Kevin has done is he's set up some high throughput screens to search chemical libraries for things that can inhibit CD14. So one of his assays that he developed, which he did a really nice job with, is a fluorescence polarization assay. I actually borrowed this diagram here from Kevin. For the sake of time, I'm not going to walk through how this assay works, but just very briefly, it monitors the ability of compounds to displace a substrate analog from the active site of the enzyme. So we have a fluorescently labeled substrate probe and a catalytically dead enzyme. We can detect binding to the enzyme using fluorescence polarization, and then we can also detect the loss of that polarization in the presence of chemicals that knock or compete the probe off of the enzyme. And then Kevin also used a separate assay, just monitoring enzyme activity, a more conventional assay, to specifically look for covalent inhibitors of CD14. CD14 has an active site cysteine that is amenable to covalent modification by electrophiles that might be good inhibitors. And so he did both of these assays. He used a facility we have on campus here, a very nice high-tech facility for high-throughput drug screening using a robotic system. And got some really nice results. So this is just a couple examples of some of the compound libraries that he's looked at. This is his fluorescence polarization screen. Most compounds have no effect on binding. You get 100% probe binding. But we have a small collection of compounds here that decrease probe binding down to low levels. And those are the things that we're interested in pursuing, doing follow-up studies on. And then with the activity screen, we're looking for things that have a high level of inhibition. Most compounds in the library have no effect on CD14 activity, but we detect a relatively small number that can inhibit activity reproducibly. And so in all, Kevin has screened over 50,000 compounds now collectively in these two assays. And he gets a hit rate of about 1 out of every 1,000 compounds screened, which is great. It means we have great specificity in the assay. Here's an example of one of the structures. He picked up a number of compounds with similar structures in his fluorescence polarization assay. And so right now, with these several dozen lead compounds in hand, we're going to be looking at characterizing the mechanism of inhibition, looking at whether they affect other phosphatases, whether they're truly specific, whether they work on other CD14 orthologs from fungal pathogens, doing some structural biology to try to see what they look like bound to the active site of the enzyme so that we can make modifications to improve their potency. So, so that's great. We've got a bunch of lead compounds from that screen, but we also just accidentally, through dumb luck, stumbled across some other compounds that look very promising and are much further along in the development process now. And this is done in collaboration with another colleague at Purdue, Zhongyin Zhang. He's actually the director of our Drug Discovery Institute at Purdue, and he's made his career on developing inhibitors for protein phosphatases. And what happened is he's a medicinal chemist, and so they built compounds, and they were making some compounds that they thought would be good inhibitors for a certain class of tyrosine phosphatases in human cells. And as part of the process, they counter-screened them against a bunch of other phosphatases to assess specificity. And they had one particular compound shown here, which we just published a paper on this year, that wasn't great for the intended target, but they found, because CD14 was in their panel for counter-screening, they found that they actually were very specific and had high potency already for CD14 enzymes. And so this was nice because we had been arguing for a long time, again, based on the structure of CD14, that we should be able to create specific chemical inhibitors for it. And they kind of proved that, or demonstrated that just by accident with some of the compounds they were making in their lab. So they went on to further derivatize this compound by adding various R groups at this position in the ring structure. And they've got some really nice inhibitors for the human enzymes. But they also found some, and we actually have a patent pending on this now, so I can't show you the actual structures of this, but they have a couple compounds that actually have fairly strong preference for fungal CD14 enzymes and have very little activity towards the human enzymes, which is very encouraging. And the most encouraging thing about this is that these compounds actually seem to have some biological activity already in fungal cells, which is a huge step forward. So this is again in Candida albicans, our assay monitoring hyphal development. So here's a wild type Candida albicans colony that's been grown under conditions to induce hyphal formation. You can see the filamentous hyphae here. For reference, over here is a CD14 deletion strain that's unable to form those hyphae. And when we add these two chemical compounds to the plates, we can block hyphal development, just like with a CD14 deletion strain, at reasonable concentrations. And so these are great lead compounds now that we have moving forward. And we want to now address the questions is, can we use those compounds in our infection assays, our larval infection assay and our mouse infection assay to block infections by Candida albicans? So Kevin is working on that right now. Um, and then we also need to look at, will these things block infections by other fungal pathogens, including ones that don't form hyphae. Not all fungal pathogens form hyphae during infection. And so we want to look at, is it a general, could it be a good general therapeutic agent or lead compound, or is it just specific to things that require hyphal formation? So, and again, we don't really know that the in vivo target in that assay was CD14, so we need to do some work to verify that. And then, of course, there'll be structural biology and optimization of these compounds and things like that going forward. But we're very excited about the preliminary data that we have suggesting that they could work. So I'll take just a couple minutes. I'll go through this quickly. I want to save time for questions. But I'm a biologist, by training. I'm really interested in biological mechanisms. So we've always had the kind of driving question of why is CD14 important for fungal virulence? Based on what we know about its biological functions, we don't have a good answer for why it's specifically important for virulence in fungal species. So one of the phenotypes, and I showed a data slide previously for this, is that we know CD14 mutants are hypersensitive to cell wall stress. And this is, again, caspofungin is another echinocandin drug. And when we treat Candida albicans that lack CD14 with these drugs, they're hypersusceptible to them. And that told us that, and that's not true for other stresses. It's a very specific, so this other variety of other stresses that we tried, it's very specific to cell wall stress. And it told us that CD14 likely is doing something to promote cell wall integrity in fungal species. And this may be a conserved function for CD14 in fungi. What does it do to promote cell wall integrity? To address that, we looked at cells up close using transmission electron microscopy. And one of the phenotypes that we observed is that there's a defect in CD14 mutants in a very specific part of the cell wall called the septum. This is a chitin-based structure that's assembled between the mother and daughter cell during cell division. And normally, in a wild-type cell, it's this bright bar-like structure that crosses the bud neck, or PS for primary septum. But in our CD14 mutants, you can see this structure is highly defective in shape. Sometimes it grows into the interior of the cell. And so that could be a weak spot in the cell that makes them sensitive to other cell wall perturbations like the drug treatments that you saw in the previous slide. Right, so that's a model. And then finally, you also saw the hyphal phenotype. We know that CD14 is important for hyphal development. That's true in agar plate assays, as you saw previously. But we also found that it's true in mouse tissue. In an actual infection in mice, if we look at the affected mouse tissue, we can see that wild-type cells, it's the dark purple staining here that stains the fungal cells. You have these elongated hyphal structures in the wild-type infection, but the strain that lacks CD14, when we can even find these cells in the mouse tissues, they're always those round yeast cells. They don't form hyphae. So are these two phenotypes, the cell wall integrity and the hyphal development phenotypes, connected somehow? We never had any previous way to connect those two, but and this is very preliminary data collected by another PhD student in my lab, Michelle Lehan, and it is looking specifically at the septum using a fluorescence probe called calcium-4 white in hyphae of wild type and CD14 mutant cells. So the septa show up as these bright bars in the wild type hyphae. And what she has observed recently is that those often fail to appear or stain in the mutant hyphae. And so there are spots where we can see that hyphae should be forming, and they're not in the mutants. And so it's possible, again, that this represents a cell wall defect, specifically in hyphal cells, that may prevent the hyphae from extending fully and forming, and could explain the loss of virulence. So that's a preliminary hypothesis we have now, but it's one that we're excited about pursuing that might provide a single nice explanation for why this protein is so important in multiple fungal species for being able to establish an infection in their host organisms. So that's it for the data. Again, we're moving forward. We're excited about exploring these phenotypes further and understanding more about CD14 biology, looking at our lead compounds in infection assays, and doing structural biology to further optimize these inhibitors to the point where they might be useful in actual preclinical drug studies, and then using our oxygen degradation system as a functional genomics platform to identify other drug targets that we can then pursue as well. So with that, I'll skip over the conclusion slide, and just again thank. This is my current group of graduate students here on the lower right. Justin is a co-supervised graduate student I have with a colleague here, Scott Briggs. A number of undergraduates are working on these projects as well. And as you saw, a lot of this work is collaborative. We have a great group of folks here that are helping out with this project at Purdue, the Chemical Genomics Facility. We have some external collaborations on this project as well, and are fortunate to have funding, a lot of funding right now from the National Institute of Allergy and Infectious Disease to support this work. And so with that, I will stop the presentation, and I'll be happy to answer questions. And thank you all for your attention, and thanks again for the invitation.
So thank you very much, Dr. Hall, for that wonderful presentation. Very exciting. And for all the data, and congratulate you for the data and the work that you have done in your work. So yes, let's go to questions. I see that there's people writing. Okay, there's a question from Sasha Colon. Have CDC14 inhibitors been studied on biofilms?
No, not yet. That is a really great question. And yeah, so I mean, for audience members that aren't aware of this, so biofilm formation is a major problem, major therapeutic problem. Candida albicans and other Candida species form tough biofilms during human infections, and they are highly resistant to existing therapeutics. It's very challenging to get drugs into them. And we have not looked. We don't actually know yet if CDC14 itself, if loss of CD14 function affects biofilms. I've talked with some of my students about setting up assays to look at that. We just haven't gotten around to it yet, but it's definitely on our high priority list of things to do. And then, yeah, if we find that CD14 is actually important for their formation, then certainly the next step would be to look at the inhibitors we have and see if they can affect biofilm formation. Great question.
I don't see any other question, but continue on that thought. I see many people that have studied different um materials for different surfaces that can affect biofilm. I think that it can be very interesting to see how those surfaces that are affecting how the biofilm is generated, also how can your drugs can affect that biofilm in that biofilm that's being created on that surface. I think that will
Yep.
be very interesting also, just as I know.
Yep.
So
Yep.
let's see,
Absolutely.
any other questions?
I hope that wasn't too high level and complicated. Don't be shy asking questions. Any question is a good question.
Okay. Another question from Sasha Colon. Any new strategies for preventing infections associated with medical devices?
Yeah, it's another great question. Again, biofilms are one of the major problems. I think, as Emmanuel was noting, biofilms forming on implanted medical devices is a major issue. I've seen, so we're not doing anything with that. I've seen some people propose ideas for things that are specifically targeting biofilms, and so I think there's a lot of interest in understanding, the molecular pathways that drive biofilm formation, so that um, they can be specifically targeted. I'm not aware of any therapeutics that are working that specifically are good against biofilms or against things growing on medical devices. But yeah, so I think people have good ideas out there. I'm just not sure if anything is at the point of actually being usable yet. But it's a very important issue for sure.
So I have LSL Rovira Diaz that raised his hand.
Hello, this is
LSL.
Eli.
Yes. Okay.
Yep, you hear
Hi.
me?
Yes.
Yes.
Yeah, this is Eli from the CDC. I was curious to know whether you quantified the fungal load in the mice experiment where the mice were infected with the mutant or the wild type.
So we did not do like a traditional fungal burden assay yet. Moving forward, especially when we do the analysis of our drugs, we're going to be including that. But what we did a lot of histology. So we saved the organs and we made sections, and we, as you saw at the end there, we stained them to look for fungal cells. Just anecdotally, it's completely non-quantitative, but we have a very hard time finding the CD14 mutant cells in the mouse tissues. That was the one good slide actually we had where we had a cluster of the mutant fungal cells. So, we expect that what's happened is those are being cleared very effectively by the mice, and so they're not persisting to the end of the assay when we collect the organs. But we haven't yet actually directly measured the fungal burden. I mean, we have such a strong effect on virulence. We didn't really have any motivation to need to measure fungal burden, but we expect the effects of drugs, for example, to be a little more subtle, perhaps, and so we will definitely be looking quantitatively at fungal burden in those experiments.
Okay, thank you.
Yeah, thank you for the question.
So any other questions from the group? I just want to make a quick question. That I just wanted to know, what are the key challenges in finding these new drug targets in fungal pathogens, and how can we overcome those challenges?
Yeah. I mean, that's like the $1 million question. So I don't know if there are any great answers for it. I mean, I think one of the real challenges is getting, and this is Sasha's question about biofilms, get to this. One of the real challenges is getting drugs to the sites of infection in the body and having them be effective, getting into all the cells and killing them. So I think it's not that hard to come up with compounds that will inhibit an enzyme, but come out with ones that have good bioavailability, good drug-like properties, in a system, can get to the sites of infection in the body and work effectively there. It takes a lot of time and effort. I mean, that's, drug companies that are doing that, it typically takes them, 10 years and, billion or $2 billion to get through that whole process. So, that's a real challenge. And there are some new antifungal drugs in clinical trials now that have new modes of action. So hopefully we'll see if some of those, work out. But it's been a long time since we had I think the echinocannins were introduced in the early 2000s, and we haven't had another, really effective antifungal drug class appear since then.
Okay. So yeah, I think we're all good with today's presentation. Dr. Hall, again, we are very thankful for your time, for the presentation. It was very, very good. A lot of very new information for us to look at. So thank you very much, everyone, for being here again. Just to remind you, we have other presentations next week on the 1st and the 3rd of October. Please attend. We have just also as a reminder, we have the semi-annual meeting on October 26th. The registration is already open, and the call for abstracts is also open for other for different students, or if anyone wants to present their poster, their data. So again, thank you, Dr. Hall, for your time. And
Thank you for the invitation. It was a pleasure.
I hope we have you presenting another time soon. So
All right.
again, thank you very much, everyone, and have a nice evening.
All right. Thank you.